Hi guys, I'm Swati. Welcome to Micrograsp. India, one of the largest producers of spices, once used to top in the global market in the export of spices. But the scenario is slowly changing. Do you know why? Yes, it's because of decline in the flavorful taste of spices due to adulteration. What is adulteration? Adulteration is nothing but any addition or deletion of substances to the original product which can change the quality of the product or its natural shape. Adulteration diminishes the quality of the food. Also, it is unethical as per the food safety and standards. Adulteration was recognized in 1950 in ancient Rome and in Middle Ages and until date. It's also important for us to know the adulterants are being used. Adulterants like simple stones in rice to harmful boric and wheat powder and something else. An adulterant is a substance which is used in altering the food. Adulteration can occur in any of the stages like from processing to the selling. There are different types of adulteration like intentional, incidental, natural, metallic and packing. Let us see few examples of these adulterations. First one is intentional. Intentional is the one which is done intentionally by addition of substances or deletion of vital parts from the original product, which causes serious health problems to the humans. Example include water and excess moisture to the milk and milk products. Also, some sweeteners are added to the honey. Talcum and chalk powders in rice flour, wheat flour and some other flours. Next is incidental. Adulterants found in food substances due to carelessness, negligence and lack of improper storage. Examples, dropping of rodents and larvae in food. Also, bacterial and fungal contamination can cause adulteration, like poor agricultural practices like harvesting, processing and storage conditions can facilitate this bacterial and fungal contamination. Natural. Presence of chemicals or other organic compounds that naturally occurs in the food but which is not intentionally added, which causes serious problems to the humans. Example include toxic variety of grains, mushrooms, green vegetables, fish and seafood. Metallic adulteration. There are certain metals at which after certain concentration are injurious to health. This can be either natural or synthetic. Example, arsenic from pesticides, lead from water. Packing hazards. There are conventional materials which are still in use like tins, jars, wax paper, cardboards, carton boxes, etc. Adulteration, though one way it is beneficial to the producers or manufacturers, it has a long-term health impacts on consumers like impurities in food, lessens nutritive value, reduces the taste, harbor disease-causing microorganisms to gastrointestinal tract, cancer, insomnia, joint pain, diarrhea, dysentery, vomiting, abortion and many other diseases. As per the response of our earlier video, Sneha had shared a video with us in adulteration of honey. So let's see how it is done in the lab environment.
Hi, I'm Sneha. Today we are discussing about adulteration of honey samples. Now, what is adulteration? Adulteration is nothing but intentional addition of substances to the honey sample for monetary gains. For example, liquid glucose, fructose syrup, sugar solutions, etc. Today we are performing for the Feast test. Feast test is performed for the detection of added sugar in honey samples. For this we require 2 test tubes, honey sample, diethyl ether and 1% of resorcinol and also a pestle and mortar. Let's see how Feast test is conducted. For this we require 2 test tubes. In one test tube I am going to add honey solution. and in the second test tube another honey sample. Next, we are going to add diethyl ether solvent to each test tubes. After adding diethyl ether, you need to swirl the sample slowly. Now, we are going to add 1% of resorcinol solution to each test tubes. And after addition, you need to shake it vigorously. Like this. And keep it aside. Okay. After the addition of resorcinol, we need to shake the test tube vigorously. So after we shake the test tubes, we will keep it aside for few seconds. Now you can see in one of the test tubes there is an appearance of cherry red color. This means the sample has been adulterated with the added sugars. And in the another test tube there is no cherry red color. So that means there is no added sugars. The appearance of cherry red color indicates the presence of HMF. HMF stands for hydroxymethyl furfural, which is present in high concentration in sugars. So when the HMF content reacts with the resorcinol and diethyl ether, it forms a complex in this color, that is cherry red. So that indicates if the honey sample turns cherry red color, that indicates it's been added with sugar. If there is no cherry red color formation, that means it is not added with sugar.
So we have seen the lab testing process of honey. Let us see some techniques which can be done at home of few products. At home, honey can be tested by adding it in a glass of water. Pure honey will settle at the bottom as lumps, whereas the other one will dissolve in water. One more example is chili powder. When you dissolve chili powder in a glass of water, the fake one will leave color. The other one will settle without leaving a color. Since prevention is better than cure, some precautions can be taken to avoid food adulteration, like purchasing in a reputed shop, checking for date of expiry, manufacturer and source of the product, the intactness, and also check whether the shop is inspected by food inspectors in a periodic manner. Rate of adulteration is twice higher than national average of 19% given by Food Safety Standards Authority of India. Presence of microorganisms in fresh foods and ready-to-eat foods can be said as the food is adulterated. But if the pathogenic microorganisms are present and can be killed by cooking or at higher temperatures, then the food is not said as adulterated. Hope you guys liked the video. Do share your lab experiences with us so that we can include in our upcoming videos. Please like, share, subscribe and comment. Also follow us on Facebook and Instagram. Until then, keep loving and learning microbiology.